So what Sutton did was, in order to prove the chromosomal theory, he worked on grasshopper. First, in the female reproductive system, that is the gynoecium in the case of plants, the embryo will be formed and that later on will develop into seed. Thomas Hunt Morgan, he studied the chromosomal theory or chromosomal inheritance on Drosophila flies. Hello everyone, a warm welcome to today's session on the principles of inheritance and variations. I'm Dr. Divya, Biology Faculty, Vidhyaashram Pre-University College, Mysuru, Temple of Excellence. So in the previous session, we had discussed about the monohybrid crosses, wherein Mendel, he selected the pea plant having only one character and only one gene representing them, be it either the tall plant or the dwarf plant, which has only one character each for tallness and one is for dwarfness. And the second experiment that he conducted was on the color of the pea flower, wherein he chose one pea plant producing purple color or violet color flowers and the other pea plant producing the white color flowers. So only one character or trait he considered for his experiment, and he crossed those two plants having one character or trait, and therefore he called it as the monohybrid cross. But in today's session, we shall deal with the inheritance of two genes. So far, we had only studied about the inheritance of one gene, wherein we studied about the monohybrid cross. But in today's session, we shall deal with the dihybrid cross. Di means it is two. So two genes or two characters which are controlled by two different set of genes are studied in this particular session. So what Mendel did in order to conduct his experiment, again he selected two plants. So before selecting two plants, what should he do? He should emasculate one of the flower, right? What is emasculation? Wherein the anthers are removed. So he'll remove the anthers of one of the flower and he will retain the anthers and the stigma of the other flower. So that flower wherein the anthers are removed, it will become the female flower, and the other flower having both the anthers and the stigma will be considered the male flower. Why didn't he remove the anthers in one of the flower? In order to prevent self-pollination, because if self-pollination occurs, then no variation will occur in the F1 generation or the offspring. So for that, so after he did that, he made the two plants to cross-pollinate. So wherein for today's experiment or for the dihybrid cross experiment, wherein he wanted to study about how the offsprings inherit or carry two genes or get two genes from the parents. So what he chose was, he chose a pea plant which is having a round shaped seed, and one more pea plant he chose which is having yellow colored seeds, and it is yellow colored seeds produced by one pea plant, that is the parental pea plant. It was round in shape and the color of the seed was yellow. So the other pea plant, consider it to be the female. He chose a pea plant, female pea plant which produced seeds which were wrinkled and it was green in color. So these two, and he wanted to see whether the genes will express themselves separately in the offsprings or not. So for that, he conducted an experiment and we shall see about it. So what he did was, he represented the round shaped yellow colored seed as capital RR and capital YY. Why it is RR and YY and not just R and Y? Why it is not R and Y? And why is it RR and YY? Because always they occur in pairs. You should represent it in alleles. So genes, they have two alleles. So that is why it is written in pairs like this, RR and YY. And what is dominant and recessive? Dominant means it will suppress the recessive character. So here dominance is always represented by the capital letters and recessive characters are always represented by the small letters. So whenever there is a dominant character character and a recessive character, it is the dominant character which will get expressed. The recessive character will not get expressed. And similarly, if both are recessive, then the recessive character will get expressed as such. So all this you have studied in the previous session. So we shall move further and see how exactly Mendel conducted the experiment, what a set of plants he crossed and what result he gotten. So moving on to the experimental part. So what Mendel did was he took two pea plants. So having two different characters. What is the, so we'll consider this to be the male pea plant and this to be the female pea plant, and they are the parental generation. So they are the parents. So they are the parents. So he selected them and he made a cross between them. Here you have to remember it is a dihybrid cross. So two characters were selected. What were the two characters? In the male parent plant, he saw that the male parent plant produced round seeds. So you can see here the shape of the seed is round. It produced round seeds and yellow in color. So it is dominant, round seeds and yellow in color, which is represented as RRYY. And next he made a cross with the wrinkled green seed. So the seed, you can see it is wrinkled, it is not completely round, it is with creases, it is wrinkled and green color, and it is the recessive character. So it will be represented by RRYY. So he made a cross. So when he made a cross, what are the gametes that are there here now? We will just see. So in order to make a cross, the gametes are needed, right? You know that the male gamete, which is the male gamete there, the pollen grain, the microspore, which you have studied in your previous sessions or you have studied in the chapter Sexual Reproduction in Flowering Plants. The male reproductive structure, that is the male reproductive gamete, the male gamete, it is the pollen grain, and the female gamete is nothing but the ovule, right? Ovule. So what are the gametes that will be present in the pollen grain as well as the ovule? So what gametes will be present here? What are the gametes that are involved here? It is RY and RY. So what are the gametes that are involved here? RY and RY. Along with that, again RY and RY. So these four types of gametes are involved in this. So RY, 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 RY. Again here RY, RY and the RY again. So four types of gametes. Now in order to see what the F1 generation plants will look like, what the characters they will carry, we will put a Punnett square and we will calculate. So for the F1 generation, we shall see with the Punnett square. So I'll put a Punnett square and we will see what will happen here. So we have RY, RY, RY. So when a cross is occurring, what can we expect in the F1 generation? R, Y, Y. Again here, Y, Y, R. We are crossing R, Y, Y, R, Y, Y. Again R, capital Y, small y. Again R with R, capital Y, small y. So what is the seed here now? The color here, this particular F1 generation plant is carrying the dominant gene R, the recessive gene R. Which shape of the seed will be expressed here? It is round, because round is dominating. So it is dominating the wrinkle character of the seed. So the seed will be round. And the color, it is yellow. Again here, round, yellow. Again round, yellow. Here the same thing, round, yellow. So what it means? All the F1 generation will have round, yellow plants. F1 generation will be round seeds the plants will produce round seeds which are yellow in color. So that is what he got in the F1 generation. So next what he did was he made a cross between the F1 generation again. So next what he did was he made a cross between the F1 generation to get the F2 generation. So in F1 generation all the seeds were round yellow. There was no difference. So his experiment was incomplete. So what he did was again he crossed the F1 generation to see what happened. So we shall see when he crossed the F1 generation what happened. So F1 generation, he made a cross between these two. Only he selected two plants from the F1 generation itself. So now we have got the F1 generation wherein Mendel found that all the plants or the offsprings, they produced seeds which were round and which were yellow in color. So his experiment was incomplete. Again, it's a confusion. So what he did was, again the F1 generation, he crossed them. So I'll just erase this and then we shall see what will happen when a cross is made between the F1 generation, what are the gametes involved, and the F2 generation plants, how will they look, what kind of seeds they will produce, what color the seed will be, all that we shall see. So Mendel, what he did was, what are the gametes that were formed? RR, Y, Y. So all the plants were RR, Y, Y in the F1 generation. So he made a cross with the F1 generation itself. So he made a cross again with one more plant of the F1 generation. So what are the gametes that are involved here? 4 different gametes are involved. So one is R, Y, R, Y. The other one is R, Y. Then again R, small y, and R, Y. Similarly here these are involved. R, Y. So these he put them in the Punnett square and he made the calculations to see what will happen, what the progenies will look like in the F2 generation. So in the F2 generation, what we will get, we shall see. So when he made a cross between these, so RY with RY, then capital RY, the dominant R with the recessive Y, and dominant R with the recessive Y, what happens, we shall see. So here they produced round yellow seeds. The seeds were round and yellow. Again here the seeds were round and yellow. Here also the seeds were round and it was yellow. Similarly here, round seeds, yellow color. Round seeds, yellow color. Again here, round seeds. So now you can guess what will be the seed color here. It will be green. Why? Because both the recessive Ys are there, recessive alleles are present there, which is representing the green color. So therefore recessive character itself will be expressed here. So you have the green color. Then again here round, yellow, round, green. Then again here round, yellow, and round, yellow again. Here what the shape of the seed will be? It will be wrinkled because it is carrying the recessive allele for the wrinkle character, right? So it will be wrinkle here and yellow in color. Again here wrinkle, yellow in color. Again here round and yellow in color. Here round, it is green in color. Again here it is wrinkled but yellow in color. Here it is wrinkled and it is green in color. So we shall see now what is produced in the F2 generation. So in the F2 there is the progenies, that is after crossing the F1 generation, the same 2 plants of the F1 generation, what the progenies in the F2 generation will look like. There will be plants which produces round yellow seeds, round yellow seeds, and there will be plants which produces round green seeds, green colored seeds. Plants with wrinkled, plants will produce seeds which are wrinkled and yellow in color, and also the progeny, in the progeny there will be plants which will produce wrinkled seeds, wrinkled, which are green in color. So we will find out the ratio now. So I told you in the previous session, in the ratio, it is of two types. One is the phenotypic ratio and the genotypic ratio. Phenotypic ratio is nothing but the morphology, external structure. Just when you see the plant in your naked eye, how will the plant appear to look? What color it will be? Say for example, in this case, we are talking about the seed. So, pea plants, when you look at the seed, you can say whether it is neatly round, whether it is yellow in color. So, that is nothing but the phenotypic ratio. So, what will be the phenotypic ratio? Just by looking here, we will understand how many yellow round seeds are there, which are yellow in color, how many round seeds are there, which are green in color, and how many wrinkled seeds are there which are yellow in color, and how many wrinkled seeds are there which are green in color. So we shall see. So how many round seeds are there in the F2 progeny? How many plants will produce seeds which are round and yellow in color? We have 1, 2, 3, 4, 5, 6, 7, 8 and 9. So this is 9. So, in the F2 generation, the progenies, how many plants are there which will produce round seeds which are green in color? So, round seeds which are green. So, you have 1, then 2 and 3. So, 3 seeds. So, 3 seeds. 3 seeds are there. So, how many? In the F2 generation, progenies, how many plants are there which will produce seeds which are wrinkled and yellow in color? We shall see. So, wrinkled means it will be small r, right? So, wrinkled yellow, 1, here 2, wrinkled yellow, 3. So, it is 3. Again, in the F2 generation, how many progenies or plants are there which will produce wrinkled seeds which are green in color? So, how many? Wrinkled and green. So, nowhere, only this is there. 1 is there, wrinkled and green. So, that is 1. So, this is the phenotypic ratio. So, now we will move on to the genotypic ratio. So, genotypic ratio is nothing but in a particular gamete. So, in the F2 generation, what are the gametes that contain the alleles or the genes which are responsible for the production of these different colors of seeds and shape of seeds? So, we'll concentrate on the genotypic. So, this was the phenotypic. So, in the genotypic ratio, we shall see. So, how many RR, YY is there? How many RR, YY is there? RR, YY. And again, how many RR, YY? So, I've taken this RRYY, then this RRYY, and again this I've finished, and again RR, this already it is done. This is there, RR capital Y small y, RR capital Y small y. So, RR capital Y small y, this is done. Then this already written here, RR small YY, it is not done. Capital R small YY, capital R small YY, and this is done now. And RR capital Y, it's already written. RR small YY, it is not written. Yeah, it is written already, RR small YY. So, I have considered already this, and RR capital YY, it's already written. RR capital Y small y, it's already written. So, small r capital YY, not written. So, I'll consider this small r capital YY, and we have small r capital YY, small r capital Y small y, it's not written. Small r, this one not written, this one I've taken. Small r, r, y, y. So like this, we have RR, YY, YY, RR, YY already taken, RR, YY. We have RR, YY. So how many? This is the genotypic ratio now. So based on the alleles, we are counting. So how many are there? RRYY, only one is there. RRYY, only one is there. Nowhere there is RRYY. So 1 is to how many dominant R genes or dominant R alleles and 1 dominant Y and 1 recessive allele. Again, it is 2 are there. So this again 2 are there. So we'll count where 2 are there. So one is this one, RRYY, and the other one is RRYY. So this one. So 2 are there. 2. Next we have dominant R, recessive R, dominant Y and recessive Y. How many are there? 2. So we'll search for them. RRYY. So RRYY and RRYY. So this is again 2. Next we have capital, that is dominant R, recessive R and 2 recessive Y. How many are there? Dominant R, this one, 2 recessive. So how many are there? We will just see. So dominant R, YY, only 1 is there. Dominant R, YY, only 1 is there. So like this, we will calculate the, what is that? Genotypic ratio. So this is how genotypic ratio we will have 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. So 4. So 4 means it is R, dominant R, recessive Y, dominant Y, recessive Y. So we'll count here. Dominant R, recessive R. Dominant Y, recessive Y. How many are there? So we have here dominant R, recessive Y, 1. So dominant R, recessive Y. Dominant R, recessive Y, second one. And again dominant R, recessive Y, third one. Again dominant R, recessive Y, again fourth one. So 4 are there. So 4. That is how the calculation is done for the genotypic ratio. So this is one important experiment which also proved the law of independent assortment. So this character that Mendel considered for his experiment, which is called as inheritance of 2 genes, wherein he went in for dihybrid cross, it is also applicable to one of his laws, that, or in other words we can say that based on this experiment, that is dihybrid cross experiment which Mendel conducted, based on this he put forth he put in a third law which is called as the law of independent assortment, which is Mendel's third law. So in the previous sessions you had studied about Mendel's law of dominance and the law of segregation, right, which are called as the, so all the 3 laws are together called as the law of inheritance. So under law of inheritance you had studied law of dominance and law of segregation. Here we will study law of independent assortment. So what does law of independent assortment states? It states that when there is more than one pair of characters is involved in a cross or involved in cross-pollination, what happens? The factors. So what are the factors according to Mendel? They are the genes. So the factors or the genes, they independently segregate and express themselves in the progeny. So here they have independently segregated, right? So according to his experiment, wherein he used the round seeds which were yellow in color, the wrinkled seeds which were green in color. So now there are 4 characters which he used here. The one parent plant produced round seeds, which is 2 characters, wherein one character was the shape of the seeds, wherein he chose the parent plant having round seeds and the parent plant having wrinkled seeds. So the second character that he chose was color of the seeds, wherein he chose one plant having the green color seeds and the other plant having the yellow color seeds. So that is why it is called as dihybrid. So when a cross was made, all the characters got expressed separately. So the shape of the seed, the alleles which was responsible for the shape of the seeds got segregated or got assorted and it got expressed separately, and the color of the seeds got expressed separately. So that is what is meant by the third law of Mendel, which is called as the Mendel's law, that is law of independent assortment. It is called as law of independent assortment. So his law nobody accepted. So it was in 1860 when he conducted experiments on the pea plants, and he put all the three laws, that is the law of dominance, the law of segregation, and the law of independent assortment. But the people during those years, that is in 1860, they were not ready to accept whatever Mendel had done. At that time, two scientists or three important scientists, Hugo de Vries, Correns and Heinrich von Schirmer, they in the 1900s, so Mendel conducted his experiment in 1860. But these three scientists, that is Hugo de Vries, Correns and Heinrich von Schirmer, in the year 1900, they rediscovered Mendel's results. So people were not believing on what Mendel did. So we shall discuss why people did not believe on Mendel. We shall discuss later. But before that, thanks to these three scientists, because they were the one who introduced Mendel's or rediscovered Mendel's experiments, and they then introduced into the world. So what these three scientists they did was they worked again on Mendel's three laws, that is especially the law of independent assortment. They worked on it. Why? Because during the time of these scientists, that is in the year 1900, the microscopes had advanced, that is the technology that was used in the microscope was advanced, and the scientists were able to study the cells. So they found out that inside the cell there is a nucleus, and inside the nucleus there is the chromosome, and in the chromosome the genetic material, that the DNA is present. So DNA or the genetic material is present inside that. So that is when they started observing the characters and found out that whatever Mendel said, it is correct. So that is why it is very important. These three scientists are the ones who introduced or reintroduced Mendel's work to the world in the year 1900. So until then, until 1900, though 1860 itself Mendel had conducted all the experiments, people did not believe him, and therefore his work was suppressed, or it was not published, or it was not made known to the people. And these three scientists were the ones who made or proved that whatever Mendel said was correct, and they made the people believe in these things. So I told you about the experiments that was conducted by these three scientists, Hugo de Vries, Correns and Heinrich von Schirmer. So they studied the chromosomes, and they found out that there are certain factors that are present in chromosomes. So earlier it was called as factors, and they said that those factors are called as genes, and genes are the ones which bring about variations or segregation of characters in the parental gen, or in the progenies, or in the offspring, or in the F2 generation. So they introduced a theory which is called as the chromosomal theory of inheritance. So chromosomes, the genes present in the chromosomes. So we inherit our parents' chromosomes. So what is there in the chromosomes? DNA is there. So what is the DNA? It is the genetic material. What is there in the DNA? Genes are there. That is how through the chromosomes the genes are transferred from the parent to the offspring. So that is why it is called as chromosomal theory of inheritance. We are inheriting the characters from the parents to the offspring through genes which are carried by the chromosomes. So when the chromosomes during cell division, when a part of the chromosomes will be divided and it will be passed on to the cells. So that is how the exchange of genes take place. So what was the reason? So I told you nobody believed in Mendel's work until 1900s. In spite of he doing all the experiments and his results proving right in 1860, they did not believe. So why did the people did not believe? It is for the following reasons. Why? Because during those days communication was not easy. People were not ready to think beyond their ethics or think beyond their religious beliefs. They didn't want to believe the scientific ideas. So that is one reason. Communication was not at all easy. That is why Mendel's work did not get recognized in the 1860s up to the 1900s. And also Mendel's mathematical approach. So I had told you Mendel, he had a binomial expression. He used the mathematical binomial expression to prove how many plants in a population have a specific character or not. So wherein he used a formula ax plus by whole square, right? And not just that, he always represented the characters in the form of numbers. Say for example, he represented phenotypic ratio 9:3:1, genotypic ratio 1:2:1:4 like that. So that is also one of the reason. Using of mathematical approach was one of the reason which people did not want. People were not ready to mix mathematics with science. So that is also one of the reason why his approach was laid back. And also the concept of genes, which Mendel was called it as factors, they were not stable and they were discrete until, and this was not accepted. Why? Because in a population of plants, it is not that only certain genes will be carried. Sometimes there will be a mutation or something which will result in the progeny looking something different than the parent. The best example is the codominance. If you remember the codominance which we had studied in the previous sessions. So in codominance, what happens? Both the traits will mix together. That is, one experiment was conducted on the snapdragon flower, right? Wherein they used the red color flower and the white color flower. And both the red and the white, they were not getting suppressed, or they were not dominating or suppressing each other. They were co-dominating each other, and both the characters got mixed up and got expressed as a pink color flower. So that is the reason. Okay, red and white. So according to Mendel, what did he say? If a cross is made between the red color flower and the white color flower, in the progeny or in the F2 generation, the red and the white will get expressed separately. But what happened in nature when a cross was made between the red and the white color flower? In the case of snapdragon, a pink color flower was produced. In the red color flowers, plant producing red color flowers were also there. Plants producing white color flowers were also there. And apart from that, there were a set of plants which produced pink color flower because of codominance. So therefore, his theory was not that acceptable for the people because of this. That is why they started saying that genes are not stable. There are times when there is codominance occurring. So that is one of the reason. And Mendel, he could not provide any physical evidence. So the other three scientists, Hugo de Vries, Correns, Heinrich von Schirmack, they were able to provide evidence because microscopes and all were advanced during the 1900s. But when Mendel was working on pea plants in 1860, there was no microscopes available, no advanced microscope, wherein he could not study about the chromosomes and all that. He just made phenotypic observations and he calculated. So that is why that evidence what he gave was not satisfying to the people. Mendel, he could not provide any physical proof or evidence for the existence of factors. So he could not provide that, he could not say that there is a cell, inside the cell there is a chromosome, in the chromosome there is the DNA, and the DNA carries the genetic material. He just said there is something called factors which is carried from the parent to the offspring because there was no microscopes. But now those three scientists, they did microscopic studies and they found out that it was the genes that are present in the chromosome wherein the DNA, that is the hereditary material, is there. They get transferred from the parent to the offspring. People were not ready to accept his. So those were the three scientists who proved that whatever Mendel said was correct. So I will just tell you. So what happens here is chromosome is the hereditary material. So chromosome, it is the hereditary material and it is carried. And what did Mendel say? Mendel say the gametes are carried or the alleles are carried. So both are same, what he said. But he didn't have a proof there. And what did the scientists in the 1900s, they said was, the genes are carried. That is what Mendel also said. The gametes are carried. And they also said that the chromosome occurs in pairs. That is what Mendel also said. The alleles, the chromosome, the genes have alleles which are also in pair. That is why we represent it as R, R, YY, right? In pairs, we represent it in pairs. But Mendel called them as factors or alleles. But after the advent of the microscope, those three scientists called it as the chromosomes. Chromosome occurs in pairs. So it was matching with whatever Mendel had said, because microscope was not there and no evidence was there for Mendel's work. People did not believe it. And apart from that, there is one more scientist, that is Walter Sutton and THEODORE BOVERI. These two scientists also conducted further experiments on those other three scientists. So, other three scientists, Hugo de Vries, Correns and Heinrich von Schimmang, what experiment they had conducted? Walter Sutton and THEODORE BOVERI, they also conducted experiments and further put strong evidences stating that whatever Mendel said is actually correct, and Mendel therefore became the father of genetics. So what Sutton did was he worked on animals. So earlier the Mendel and the other three scientists, they worked on the plants to prove the theory of inheritance. So what Sutton did was, in order to prove the chromosomal theory, he worked on grasshopper. So when he worked on grasshopper, he also found out that from the parents the traits are carried on to the offspring. And Boveri, he worked on sea urchins. So when he was working on sea urchins, he found out that in the embryo, if the embryo has to express the characters of the parent, all the traits should be present in the gamete. So all the chromosomes should be got from the parent and it should be present in the gamete if they have to be expressed in the embryo, later on expressed in the offspring that is developed. So that is what they said. So what Walter Sutton and THEODORE BOVERI, in their chromosomal theory, what they said was, after their experiments on the grasshopper and sea urchin respectively, what they found out was, they found out that the chromosomes, first they will be united and later on they will segregate. So Sutton, what he did was, he united the chromosomal segregation with Mendelian principles and he called it as the chromosomal theory of inheritance. So whatever Mendel did, that he compared with his experiments or his results that he got while working on the chromosomes of the grasshopper, and he said that whatever Mendel said in his theory or in his laws, that is especially the law of independent assortment, all that is correct. And Sutton, he described or he put forth the chromosomal theory as the pairing and separation of a pair of chromosome. So the pairing and the separation of a pair of chromosome, it will lead to segregation of pair of factors they carried. That is what Mendel also said. Mendel said the characters will get segregated, the alleles will get segregated and they will get represented individually in the progeny. That is what these scientists are also seeing in the chromosomal theory. So in the chromosomes, first the chromosomes will be paired and during cell division they will segregate, and each chromosome, what characters they are carrying, it will be segregated, it will be assorted and it will be carried to the next generation. That is what they are saying. So that is how all the 5 scientists, they worked on keeping Mendel's experiments as a base. They worked on it and they proved that whatever Mendel said was exactly correct. So we should pay our tribute to all these scientists, and because of them, Mendel got into fame. People started believing Mendel and keeping Mendel's experiment as a base. They started doing all the other genetic experiments, and that is why Mendel was the first person to prove that there are some factors called as genes which are carried from the parent to the progeny and they get expressed in the progeny. So therefore, Gregor John Mendel, he is called as the father of genetics. So what is this chromosomal theory exactly? So we know that there is a cell. So we'll take the example of a male gamete and a female gamete in the case of a flowering plant. So in the flowering plant, the male gamete is the pollen grain. So we'll take a male gamete, that is the pollen grain, and we will take the female gamete. So these two will fuse. So when they fuse, what happens? The progeny will be produced. First, in the female reproductive system, that is the gynoecium in the case of plants, the embryo will be formed, and that later on will develop into seed, and that seed is the one when sowed gives rise to the new progeny. So what happens in the progeny? Now the parent will have one cell, and also here there is a cell. They will have their own set of chromosome. They will have their own set of chromosome. So these chromosomes, they will be carried to the progeny. So progeny. Say for example, in the male plant, they have a white color flower. So white flower. And female plant also has white flower, but there is one more character, that is, it has green leaves. So here what happens when the crossing is done? They will get expressed separately. So in the resulting progeny, all the cells will get expressed separately. So this is white flower will also occur in the plant, and also the green leaves will also occur in the plant. So that is nothing but chromosomal theories. Here the characters are separated. They are carried as it is to the progeny, and they are all the characters will get expressed in the progeny depending on whether they are dominant or recessive. So they will get expressed separately. So each character will get expressed separately. So in the plant, in the resulting progeny, what happens? The white color, the flower will be white in color. The color of the leaf, which was supposed to come from the female parent, that also got expressed. So that is how each character gets expressed separately. And moreover, you have to understand depending on whether it is dominant or whether it is recessive. So here you can see there is one cell. So in the cell, the chromosomes are there. In the next stage, what happens? The duplication, that is during meiosis, the duplication of the chromosomes will take place. So once the duplication, they have doubled in their number. So once the duplication takes place, they starts to enter into the, first is the interphase phase. So in the interphase phase, what's happened? The cell prepares itself for the duplication of the chromosome. So in the G1 phase, you have the G1 phase, the S phase, and the G2 phase in the interphase. So different phases of cell division. In that, the first phase of cell division is the interphase. It is the interphase. So in the interphase, there is the G1 phase, the S phase, and the G2 phase. So in the G1 phase, what happens? There is the cell prepare the chromosomes. They are not in duplicates. So the chromosomes that are there, they will prepare, or the cell will prepare themselves for the duplication of the chromosome. So as soon as in between there is a phase called as S phase. So in the S phase, the duplication of the chromosomes will start, and in the G2 phase, homologous pairs of chromosomes will be produced. So homologous pair, one homologous pair, other homologous pair. And these homologous pairs, they are nothing but the bivalent. They are bivalent in nature. And again, they will undergo meiosis, wherein they will enter into the, so before that, what happens is there is a stage called as the metaphase. So in the metaphase stage, all the chromosomes will align in one equatorial plane. So they will start to align. And after they align, what happens? These homologous pairs of chromosomes, they will align, and later on, after the alignment, they will start to segregate through the spindle fiber. So this is a spindle fiber. So through the spindle fiber, they will start to move like this. They will start to segregate. So when they are segregating and when they are entering into another cell, so this segregation, again, they will form two different cells. So they will form two different cells. So here one, the chromosomes. Here the chromosomes, they will separate like this. So this is nothing but independent. Now the chromosomes, so whatever genes are carried by the chromosome will get expressed here separately. These genes will get expressed here separately. So that is chromosomal theory, wherein the assortment or the separation or the segregation of characters is taking place. So in meiosis 1, what happens? The chromosomes, they will start to separate. They will start to move towards the two poles along the spindle fiber, and then Meiosis is nothing but reduction division. Here when the division takes place, two cells will get formed. One mother cell will get, one parental cell will develop two daughter cells. So in these two daughter cells, the chromosomes will be passed on. Again, these two daughter cells will again divide and they will form again the germ cells or the other they'll form four daughter cells, and each chromosome will enter into the daughter cell. So these daughter cells or these germ cells are the ones which are the gametes, and these will pass on their trait to the next development of the progeny or to the next progeny. So this is what happens exactly in the chromosomal theory. There are different statements that were given by the scientists for the chromosomal theory. So what are those statements? They said that chromosomes, they are vehicles of heredity. They are vehicles, right? Why? Because, for example, we carry the traits from our parents. We have some characters like brown color eyes or brown color hair. So if the father has brown color eyes and the mother has brown color hair, sometimes both will get expressed in the progeny, right? So how will they get expressed in the progeny? It is because of the chromosomes. So the chromosomes are the ones which carries the genes that are responsible for the traits that have to be developed in the progeny, and they will be moved to the progeny. They will carry the genes. So that is why chromosomes are called as the vehicles of heredity. We are getting the characters from the parents. So that is why through the chromosomes. And what are there in the chromosomes? Genes are there. So that is why chromosomes, they are the vehicles of heredity, and they are transferred from the parents to the offspring. So this is one of the statement that they put forward in the chromosomal theory. So according to the chromosomal theory, it states that chromosomes are the vehicles of heredity, and they are transferred from the parents to the offspring, and that is why the characters which are there in the parents are also expressed in the offspring. And they also said that two identical chromosomes, they form a homologous pair, as I showed you previously. So this is a homologous pair. So two identical chromosomes, they form a pair. So these chromosomes, they are different. Now two identical chromosomes, they form a pair. So that is the second character. So two identical chromosomes, they form a homologous pair. And independent pairs, those pairs which are independent, they segregate separately. So when the cell division takes place, what happens? So first they were in one cell, but in pairs. This is first pair, this is the second pair. In pairs, when the cell division took place, each pair got separated into each cell separately. That is what they express. So that is what it means. So independent pairs, they segregate independently of each other. They carry themselves forward separately into each cell, and they segregate at the time of gamete formation. Again, they will segregate in the gamete formation. So during the gamete formation, again they will segregate, and these characters separately, they will be carried to the offspring. Separately, they will move to the offspring. So this is what is meant by chromosomal theory. So this is what they meant. They said they segregate at the time of gamete formation. So they segregate at the time of gamete formation, and chromosomes, they are mutable, maybe because of exposure to radiation, because of certain environmental factors and all. The genes that are present in the chromosomes, they tend to mutate. So when they mutate, the same thing will be carried to the progeny, or the progeny may have a different set of traits in them which is not there in the parent. So that is one of the factor of the chromosomal theory. That is, chromosomes are mutable. They are able to mutate. So next, one more person, that is Thomas Hunt Morgan. So he also proved whatever Sutton and Boveri did. So after Sutton and Boveri, they performed experiments on grasshopper and sea urchin. They came to a conclusion that whatever Mendel said in his experiments are all correct. So in order to add strength to that, again one more scientist, his name is Thomas Hunt Morgan. He studied the chromosomal theory or chromosomal inheritance on Drosophila flies. So why did he choose Drosophila for his experiment? We will just see. So what he did was he chose Drosophila flies because of certain traits or characters or the easiness that is there in culturing the Drosophila fly. So what Drosophila is nothing but if you just keep a banana in your home, no, and you don't eat it for too many days, you can see a small fly like flies flying around it, which are very tiny. It is nothing but fruit fly. They are called as Drosophila melanogaster. They are the fruit fly. So why did he choose these fruit flies? For chromosome, to study the chromosomal or to prove the chromosomal theory of inheritance. That is Thomas Hunt Morgan. Why did he choose them? It is because they breed very, very quickly. That is just within 12 to 14 days, they can complete the life cycle and a new offspring can come out. So that is why they breed very, very quickly. That is why he chose that. And it has a shorter generation time. That is the life cycle of Drosophila is just 12 to 14 days. That is one of the reason why he chose them. And also he found out that throughout the year the breeding can be done. Why throughout the year? Because say for example there is an elephant. Can you breed the elephant throughout? No, only once in a few months it will give rise to a baby, and the baby or again to grow back into a fully grown elephant and then to complete its life cycle take lots of years. So that is not the case in the case that is not same in the case of Drosophila. Within 12 to 14 days again a new set of flies you can have. That is the reason. And also hundreds of progenies will develop just by mating one male and female fly. Hundreds of progenies you will get. And imagine if hundreds of male and female flies are have mated, laid their eggs and all that, then how many progenies, lakhs together progenies we will get. So that is why he chose Drosophila for his experiment. And also they can be grown on simple synthetic medium. Just squash a banana and put it in a bottle and add agar to that and just keep it like that. The flies will come, sit on it, lay the eggs, the larvae will start feeding on it, they will grow, again the flies will, it will make a home of that synthetic medium. So that is why it is very easy to culture, that is why it is used. And also the male and female flies are easily distinguishable. So the male, they are usually smaller in size, the female flies are larger in size, and also the males, their abdomen, at the end of their abdomen, it is usually round. The male abdomen, it is usually round, but the female, it is pointed. So that is, it is easily you can distinguish between them. And also females, they are large, but males are small. And one more thing is, the males, they have dark segments that are present, but females, they have light, very light segments. Not just that, even by looking at the abdominal region itself, it is enough for us to identify which is a male and a female. So easy identification. That is why he chose Drosophila, that is fruit fly, for his experiment. And also it carries many types of hereditary variation. The Drosophila, they produce red-eyed Drosophila, white-eyed Drosophila, yellow-eyed. Sometimes the wings are small, the wings are large, different hereditary characters. And it is also said that if you want to study the genetic nature or the genetic studies on humans, no need of using the human chromosomes. It is enough to study the chromosomes of the genetic material of the Drosophila, because almost 60% it is similar to that of humans. That is why. So that is one of the reason it is being chosen. And also you can just put a Drosophila on a cover, on a slide, and then observe it under the microscope. All the characters, whatever I said, the eye color, the sex comb. So males will have sex comb. So in the legs, no, the males, they will have hair-like structures like the sex combs. So all that easily it can be observed under the microscope. So that is also one of the reason why Thomas Hunt Morgan chose this for his experiment. So in the next session, we shall see how he performed the experiment and what results he got in order to prove the chromosomal theory of inheritance. So these are just the characters why he chose. He could have chosen some other animal or fly or insect, right? But why did he choose a Drosophila or a fruit fly for his experiment? Because of the following characters, or because of the easiness of all these things. That is why. So in the next session, we shall see what results he got while after conducting experiments on the Drosophila, like how Mendel crossed the male and the female pea plants, right? Similarly, a cross, that is mating, was brought about between the male Drosophila and the female Drosophila in order to study in the progeny how will they look like, what are the traits or the characters that are carried. So this was about Thomas Hunt Morgan, wherein he used Drosophila or the fruit fly for his experiment. So I hope you have understood this session well. In the coming sessions, we shall deal with the linkage and recombination. So this is one of the important things. So to prove the linkage and recombination, Drosophila is one of the best examples. So what happened and what results did the experiment put forth, all that we'll study in the next coming session. So I hope you understood this session well. We shall meet in the next sessions. Thank you.